Advotech Instructional Videos presents Experiment 303, Exploring Biotechnology with Green Fluorescent Protein. This video will outline the experimental protocol performed by the students for Modules 1 through 4. Experiment 303 has been updated to utilize the Enhanced Transformation Protocol, a new lysis buffer, and an improved staining method. Step 1. Obtain two 1.5 ml tubes of E. coli starter culture. Label the tubes with your initials or group number. Step 2. Centrifuge the tubes at maximum speed for 5 minutes to pellet the cells. Step 3. Carefully pour off the supernatant. Do not disturb the cell pellet. Step 4. Add 200 microliters of ice-cold calcium chloride solution to each tube. Gently resuspend the cells by slowly pipetting up and down several times. Save the remaining calcium chloride on ice for a later step. Note, it is important that the cells are fully resuspended. Continue to gently pipette until no clumps are seen in the calcium chloride solution. Step 5. Incubate the tubes on ice for 10 minutes. Step 6. Centrifuge the tubes at maximum speed for 5 minutes to again pellet the cells. Step 7. Carefully pour off the supernatant. Do not disturb the cell pellet. Note, at this point, the cells are fragile. Keep the cells on ice and pipette slowly and gently. Step 8. Slowly add 100 microliters of ice-cold competent cell solution, CCS, to each tube. Gently resuspend the cells in the ice-cold competent cell solution by slowly pipetting up and down several times. Immediately, place the tubes on ice and proceed to transformation. Optional stopping point. The competent cells can be stored for up to 48 hours in the freezer after they have been resuspended in the competent cell solution. Transformation with GFP plasmid DNA. Step 1. Retrieve 2 tubes of competent cells and place immediately on ice. Label one tube plus DNA and the other tube minus DNA. Step 2. Add 150 microliters of ice-cold calcium chloride solution to both tubes. Mix by gently pipetting up and down several times. Step 3. Add 10 microliters of pGFP DNA to the tube labeled plus DNA and gently flick the tube to mix. Do not add plasmid to the minus DNA tube. Step 4. Incubate the tubes on ice for 10 minutes. Step 5. Place the transformation tubes in a 42 degrees Celsius water bath for exactly 45 seconds. Step 6. Immediately return the tubes to the ice bucket and incubate for 2 minutes. Step 7. Transfer 50 microliters of recovery broth to each tube using a sterile 1 ml pipette. Gently mix by flicking the tubes. Step 8. Incubate the cells for 5 minutes in a 37 degrees Celsius water bath. Step 9. While the cells are recovering, label the bottom of 4 agar plates as follows: minus DNA, plate with no stripe; minus DNA plus amp, plate with 1 stripe; plus DNA plus amp, plate with 1 stripe; plus DNA plus amp plus IPTG, plate with 2 stripes. Step 10. After the recovery period, remove the tubes from the water bath and place them on the lab bench. Step 11. Using a sterile 1 ml pipette, transfer 150 microliters of recovered cells from the tube labeled minus DNA to the middle of the minus DNA and the minus DNA plus amp plates. Step 12. Using a new sterile 1 ml pipette, transfer 150 microliters of recovered cells from the tube labeled plus DNA to the middle of the plus DNA plus amp and plus DNA plus amp plus IPTG plates. Step 13. Spread the cells over the entire plate using an inoculating loop. Use 1 sterile loop to spread both minus DNA samples. Change to a fresh loop before spreading the plus DNA samples. Make sure the cells have been spread over the entire surface of the plates. Cover the plates and wait 5 minutes for the cell suspension to be absorbed by the agar. Step 14. Stack the plates on top of one another and tape them together. Label the plates with your initials or group number, and place the plates in the inverted position, agar side on top, in a 37 degrees Celsius incubation oven for overnight incubation, 16 to 18 hours. If you do not have an incubator, the colonies will form at room temperature in approximately 24 to 48 hours. Step 15. Visualize the transformation and control plates using a long-wave UV light. For each of the plates, record the following: the number of colonies on the plate, the color of the bacteria under UV light. Note, if possible, take a photo of the results for your lab notebook. Plating of transformed cells. Step 1. Obtain 2 LB amp IPTG plates. Step 2. Use your plus DNA amp IPTG transformation plate, result plates from Module 1, as the source of your seed culture. If necessary, you can share a plate with another group or use one of the control plates. Step 3. With an inoculating loop, pick 4 to 5 isolated GFP-expressing glowing colonies. Step 4. Spread the cells evenly and thoroughly over the entire surface of the plate. Step 5. Repeat steps 3 to 4 for the second plate. Step 6. Replace the cover onto the plates. Step 7. Incubate the set of plates in a 37 degrees Celsius incubator overnight. Step 8. Check to make sure there is a confluent lawn of growth on at least one of the plates. After overnight incubation, proceed to isolation of GFP. If additional time is needed after the 37 degrees Celsius incubation, the plates can be stored in a plastic bag at 4 degrees Celsius for up to 7 days. Isolation of GFP. Step 1. Collect a 1.5 ml microcentrifuge tube containing lysis buffer and label the tube with your initials. Step 2. Select a GFP plate showing the highest GFP expression, maximum glow, after overnight incubation at 37 degrees Celsius. Using a sterile loop, carefully scrape the entire cell growth off of the GFP plate. Step 3. Twirl the loop containing the colonies in the tube containing lysis buffer. Twirl vigorously until the cells are dislodged into the buffer. Step 4. Vortex the tube at maximum speed or pipette up and down until the cells are thoroughly resuspended. Note, be sure to fully resuspend the cells. This might require vortexing or pipetting vigorously for up to a minute. Step 5. Incubate the tube for 10 minutes in a 55 degrees Celsius water bath. Step 6. Place your microcentrifuge tube containing the GFP cells in the -20 degrees Celsius freezer for 15 minutes or until frozen. Lay the tube on its side to ensure rapid freezing. Optional stopping point. Cell lysates can be stored at -20 degrees Celsius for up to a week if needed. Step 7. After the cell suspension is completely frozen, remove the microcentrifuge tube from the freezer and put it in a 55 degrees Celsius water bath to thaw the cells. Step 8. Vortex the samples or pipette up and down vigorously for 30 seconds. Step 9. Centrifuge the tube in a microcentrifuge for 5 minutes at maximum speed. Note, make sure you have balanced your tube before starting the microcentrifuge. Step 10. At this point, the supernatant should contain the green fluorescent protein. It should be bright green under UV light. Note, if the supernatant is not fluorescent and/or the cell pellet is fluorescent, repeat the freezing, thawing, centrifugation steps, steps 5 through 9, until the green fluorescent protein, GFP, is released into the supernatant. Step 11. Transfer 200 microliters of glowing supernatant to a clean tube and label GFP cell extract. Store the extract and any leftover supernatant in the freezer for use in Module 3, Purification of GFP by column chromatography. Optional stopping point. Cell extracts can be stored at -20 degrees Celsius until needed for Module 3. Packing and equilibrating the column. Step 1. Vertically mount the column on a ring stand. Make sure it is straight and that the white cap is firmly attached to the bottom of the column. Step 2. Add 1 milliliter of 1x elution buffer to the column and confirm that the bottom cap is sealed. Step 3. Mix the slurry, molecular sieve matrix, thoroughly by swirling or gently stirring. Step 4. Carefully pipette 4 milliliters of the mixed slurry into the column by letting it stream down the inside walls of the column. If the flow is stopped by an air pocket, stop adding the slurry and firmly tap the column until the air is removed and the slurry continues to flow down the side of the column. Step 5. Place an empty beaker under the column to collect 1x solution buffer. Remove the cap from the bottom of the column and allow the matrix to pack into the column. Some fine sediment may drip through the column, but the majority will form a uniform solid matrix. Note: the matrix should fill approximately 2/3 of the narrow part of the column. If the packed matrix is too low, additional slurry can be added to raise the volume. Step 6. Wash the packed column with 5 mL of 1x elution buffer. Always keep a thin layer of elution buffer on top of the packed matrix to prevent drying. Note: the loading of the column and subsequent elution will be done at room temperature. The elution buffers and the fractions collected will be stored on ice as they elute from the column. Do not allow the column to run dry. Step 7. Slide the cap onto the spout and make sure it does not drip. Collecting column fractions of GFP protein. Safety note: wear UV safety goggles for the following steps. Step 1. Slowly load the column with 0.2 milliliters of the GFP extract you saved from Module 2. Remove the cap and allow the extract to completely enter the column, collecting the flow-through waste in your beaker. Note: if you do not have GFP extract from Module 2, you can use the provided control cell extract containing GFP. Step 2. Begin to elute the column with 1x solution buffer. Add the buffer very slowly, several drops at a time, to avoid diluting the protein sample. Remember, do not allow the column to run dry. Step 3. Monitor the progress of the GFP in the gel matrix by illuminating the column with the long-wave UV light source. It may help to dim the lights in the room. Allow the column to drain into the waste beaker. Step 4. When the GFP protein band almost reaches the bottom of the column, near the frit, start collecting the fractions in the microtiter plate. Collect 4 drops per well, starting with the first well of the microtiter plate, and work your way from left to right. Note: to prevent loss of the GFP protein, you should begin collecting fractions before the GFP reaches the bottom of the column. Step 5. Continue to monitor the progress of the GFP in the column and collect fractions until the GFP has been completely eluted. The column can then be capped and set aside. Step 6. Check the fractions in the microtiter plate by using the long-wave UV light. Identify the well that contains the brightest levels of fluorescent protein. Step 7. Transfer 30 microliters of the brightest elution to a screw-top microcentrifuge tube. Label the tube GFP native. Step 8. Transfer an additional 30 microliters of the same elution to a second screw-top tube. Label this tube GFP denatured. Optional stopping point. If time does not permit you to continue with the SDS gel electrophoresis, you may freeze the fractions at -20 and perform the assays at a later date. Preparing the protein samples. Step 1. Add 10 microliters of native protein solution, NPS, to the tube labeled GFP native. Step 2. Mix and set this tube aside for electrophoresis. Step 3. To denature the protein sample, add 10 microliters of protein denaturing solution to the screw-capped tube labeled GFP denatured and mix well. The denaturing solution contains sodium dodecyl sulfate, SDS, and 2-mercaptoethanol. Step 4. Bring a beaker of water covered with aluminum foil to a boil on a hot plate. Step 5. Carefully push the bottom of the GFP sample tube to be denatured, boiled, through the foil and immerse the tube in boiling water. Do this carefully to avoid getting burned. The tube should be kept suspended by the foil. Note: make sure the sample tubes are tightly capped and thawed if samples had been stored at -20 degrees Celsius. Step 6. Boil the GFP sample for 5 minutes. You can confirm that the GFP has been denatured by viewing the sample on a UV or blue light transilluminator or by using a handheld UV light. Unlike native GFP, the denatured protein sample will no longer glow green. Preparing the PAGE gel and chamber. Step 1. Open the pouch containing the gel cassette. Remove the cassette and place it on a lab bench with the shorter front plate facing up. Step 2. Gels may feature a sticker or tape at the bottom of the front plate. Remove the tape if present. Step 3. Carefully remove the comb by gently pulling upwards. Pull the comb straight up to prevent damage to any of the wells of the gel. Step 4. Insert the gel into the electrophoresis chamber. Orient the gel according to the manufacturer's instructions. Note: for Edvotech vertical electrophoresis chambers, the short plate should face the interior. The following diagram shows how high the buffer should be, what your electrophoresis chamber should look like when the gel is inserted, as well as how to position the pipette tip so that the samples are properly dispensed into each well. Loading the protein samples. Step 1. Using a fresh, fine-tip micropipette tip, measure 20 microliters of the first sample as indicated in Table 1. Step 2. Place the pipette tip under the buffer and directly above the sample well, resting gently against the back plate of the gel cassette. Step 3. Slowly dispense the sample by depressing the plunger. Step 4. Repeat steps 1 through 3 for the remaining samples in Table 1, changing the tip between each new sample. Step 5. Once all samples have been loaded, carefully place the cover onto the electrode terminals. Step 6. Connect the electrical leads to the power supply. Step 7. Set the voltage of the power supply and perform electrophoresis. See Table A for time and voltage guidelines. Allow the proteins to separate on the gel for the recommended length of time or until the tracking die reaches the bottom of the gel. Step 8. After electrophoresis is finished, turn off the power supply, disconnect the leads, and carefully remove the cover. The gel can now be removed from the chamber. Immediately proceed to staining instructions on page 26. Staining with flash blue protein stain. Step 1. After electrophoresis, lay the cassette down and remove the front plate by placing a thin spatula or flathead screwdriver at the side edge and gently lift it away from the larger back plate. In most cases, the gel will stay on the back plate. If it partially pulls away from the front plate, let it fall onto the back plate. Handle very carefully, as the thin gels are extremely fragile. Step 2. Transfer the gel on the back plate to a clean tray. Step 3. Add a sufficient volume, approximately 50 to 75 milliliters, of the staining destaining solution into the tray to cover the gel and back plate. Step 4. Carefully remove the back plate from the tray, leaving just the gel in the tray containing the staining destaining solution. Note, if the gel sticks to the plate, gently nudge the gel off the plate using gloved fingers. Step 5. Discard the staining destaining solution. Pour slowly to keep the gel in the container. Step 6. Add 30 milliliters of prepared flash blue protein stain. Step 7. This step is optional. Cover the container with plastic wrap and microwave for 10 seconds to gently heat the solution. Step 8. Incubate for 15 minutes at room temperature, shaking occasionally. Step 9. Discard the flash blue protein stain solution. Pour slowly to keep the gel in the container. Step 10. Wash the gel by partially filling the container with water and gently rocking back and forth several times. Discard the used water and repeat with fresh water. Step 11. Add 30 milliliters of staining destaining solution to the gel. Step 12. Incubate for 15 minutes at room temperature. Examine the gel. Step 13. This step is optional. Discard the used staining-destaining solution and add an additional 30 milliliters of staining-destaining solution. Incubate for 15 to 60 minutes at room temperature until the appearance and contrast of the protein bands against the background improves. Step 14. After staining, protein bands will appear medium to dark blue against a light background. A white light box can be used to better visualize the protein bands. To finish the experiment, use the instructions found on pages 29 and 30 to determine the molecular weight of each protein.